Okay, so this is just for a little bit of help on the mathematical modeling Hardy-Weinberg lab. So we would be using Excel, but you guys can use Google Sheets on your iPads. That will work just fine for what we're doing. So an important reminder, when you open the lab on Schoology, make sure that you make your own copy. You don't want to edit the actual lab, and it probably won't let you edit the spreadsheet that's been shared. So make your own copy that you can manipulate. If things go really wrong, guess what? Just make another copy. Okay, so first and foremost, make sure that you're reading the background. This lab isn't necessarily super intuitive, like you have to read kind of to understand what's happening. And there's not a lot that we're asking you guys to manipulate. So the small changes that you are changing are important to kind of understand what you're doing. So make sure that you're reading the background. Down here, I remind you guys that anything that's highlighted in green should be answered. You can answer these in a Google Doc would probably be the easiest thing to do. So there are questions embedded throughout the whole lab exercise. So make sure that you're paying attention to those. Read through the objectives and see what the expectations are for the lab. Make sure that you're reading all this background information, annotate it appropriately. And then, continue to read this. So we've done a lot for you, but it's important, and it will make a lot more sense when you look at the final chart if you guys are reading this background. So take your time, read the background, annotate it appropriately. So this is what this spreadsheet is going to start to look like. And when you guys open it up, you'll see that there's a lot more letters and numbers in there because Mr. Berg actually created this lab for you. So all you're doing is manipulating a few of the boxes to change the outcomes, okay? But still, it's worth reading so that you know what's happening that's already been done for you, okay? The reason that we kind of did that is because really small changes can, make error messages come up and stuff. So as long as you guys can understand what you're reading, that's kind of what we really want you to get out of this. So notice the first thing that you're entering, right, is the p-value and the q-value. So you're looking at the variation in these particular traits, right? So you're looking at these allele frequencies. The next thing that you do that we've done for you is generate some gametes. So we're randomly generating gametes. So what these are representing, like where you see an A right here, this AB, that's representing a heterozygous gamete. This next one, big A, that's representing a homozygous gamete that's homozygous for that A trait, okay? So you're making a whole bunch of gametes. And then as you keep going, you populate the zygote. So you're taking those two gametes, right? Think about the final sex cells and meiosis, and you're joining them together to make a zygote. So that's what that next column is going to show. Notice we have some questions embedded here. And then what the next part of the chart is showing is it's numbering, it's counting the amount of either homozygous for A, heterozygous, or homozygous for B that are in each row, right? So notice that big A gets a 1 right there. It's tabulated as a 1 because that zygote is represented as such, right? AB gets a 0 and BB would get a 0 too, okay? So you'll notice that we filled all of that in for you. And then at the end, you could actually make a little frequency graph. You could show what the frequency of the particular genotypes were for this one small first generation that you populated. The next thing that you'll notice is that each tab in the sheets, and I'm actually going to go forward here. I encourage you guys to read all of this, answer these questions. But what I'm talking about here is we showed you guys 4 different generations. Right, so the pictures of the lab only show you the first one. So we show you over time how randomly simulating using these P and Q values, which start with 50-50, right? There's a 50% chance of having both P and Q value, how those change over time. So some things that you want to notice, right? We didn't highlight the cells, but it's the same thing, right? We have our gametes, we have our zygotes, we have the number of gametes accumulated, and then you'll see the totals at the bottom. So you'll see the number of each allele, right? That's the allele, essentially the allele frequency, and then the allele frequency that they're going to use based on this population that was just made for the next generation. So that 0.48 and 0.52 is what was used here to generate population number 2, and then thus forward. So again, just remember to answer these questions that are embedded in here. Some of it is just really thinking about these things, okay? But when you actually manipulate it, when you actually test it yourself, what you're doing is considering what are things that could change this population. So things like natural selection, things like a bottleneck, things like genetic drift in some way, right? So think about how you could model those in your spreadsheet. So in other words, what would you change to try to find that result? Like, how would you demonstrate a bottleneck? How would you demonstrate natural selecting, maybe selecting against that A trait and for the B trait, right? What would you do? So what you're going to do, right, that first page, the standard, that you're really just looking at, right? You're using that to help you answer a bunch of questions. But for the scenarios 1, 2, and 3 on the bottom, those are the ones, so they've been copied, right? They're original copies, but what we want you to do is to manipulate them, right? And to try to demonstrate how you would, in a spreadsheet, change things to, demonstrate something that could happen in nature, right? That could cause a population to evolve. And, I would suggest even renaming them at the bottom, kind of, is it the bottleneck one, or is it some type of natural selection? So these ones you can change to fit your own personal thoughts for that. Okay, how you want to change that. Leave that standard one alone. That's your background. So to be very honest with you, there's very little that you guys have to write in. Rather than writing stuff in, you're really just kind of looking at these outcomes. Okay, I hope that kind of helps to make a little bit more sense. We do want you to, as this last part says, after you do those 3, they give you some examples, right? So you could do selection, you could talk about mutations, you could talk about migration, genetic drift. We want you to analyze your data, right? So we want you to consider what you finally get by changing a small variable in each one of those scenarios. How is that outcome over 4 generations changed? So after you guys answer all of the questions, right, it's important that you actually finalize it by, analyzing and concluding what your scenarios showed. Okay, so what should be turned in is a document with these embedded questions answered, and then finally that analysis question answered at the end.